So, welcome to one of the big experiments that I've been involved with. It's a 20,500 tree restoration experiment that's been embedded within a koala habitat restoration project. And as such, all the species we'll be planting, the royal we, are from this ecosystem, 1233, which of course includes a number of important koala food trees like Eucalyptus teraticornis and Eucalyptus cribra. When asked to sort of come up with ideas to embed an experiment into this, I'm a plant community ecologist, so it was pretty automatic that we would manipulate the diversity of the plots and we would manipulate the composition of the plots to assess in the longer term whether the identity and composition of the plots affects their productivity, how quickly they accrue biomass, which is interesting from a carbon angle but also from a habitat provision angle, and also how quickly natural processes start to kick in as well. These are the longer term goals of this project, to look at things like that. In the short term, of course, as you all know, restoration's a pretty expensive operation. It's a very uncertain thing to do as well. We like to think that we get high survival and great growth and great outcomes from all restoration projects. I think we all know enough to know that that's not always the case. So we're always looking to say, okay, what's the goal here? If the goal is to turn this from a pasture into a woodland, we've got to put trees in the ground. We want those trees to have high survival. We want them to have good vigor. So what we see in these ex-agricultural landscapes is we see soil compaction, we see nutrient depletion, we see changes to the soil microbial communities and the like. This is what ecologists call the agricultural legacy. It's a legacy of the previous land use that will live on here, I guess, for a few years until we can somehow reinstate processes that shift the balance back towards a woodland ecology, nutrient cycling and a microbial community that would be more natural, I guess. So with that in mind, in addition to the plots having different diversities and compositions, we will also be manipulating two soil treatments. The first is covered by my very inadequate tarp over here. I pulled that out of the back of my car the other day thinking, oh, that'll be good. This is soil collected from the other side of the property, from areas where Eucalyptus cribra and Eucalyptus teraticornis exist, with a really good tree litter layer and actually at the time of collection, obvious fungal fruiting bodies. So from 3 locations on that side of the property, we've gone and collected the first 10 centimetres of soil, including the litter, and we've stockpiled it here. In these plots here, I'll go through this in a sec. You can see there's pink plots and there's white plots. In the pink plots, every plant in that pink plot will receive half a cup of natural soil. Okay, so it's an inoculation. It's a very crude way of inoculating the soil. Can we simply, rather than having to use really complex nursery techniques and isolating particular fungal species, can we simply just add half a cup of what you might call nature dirt and somehow kickstart a microbial process or a microbial community? So the answer is we don't know, and we will hopefully address that with this experiment. We have a mycorrhizal expert who's already taken samples from the nature dirt pile and has taken samples all throughout this planting already. We're going to sequence those samples and see what's in them. Okay, so that's pretty neat. So that's the first thing. That gets applied to entire plots. Now in here, you will see, I'll take you down there in a sec, some plots are marked out with pink tags, and some plots are marked out with orange tags. The pink tagged plots get the nature dirt. Okay, it's pretty simple, and every plant in that plot gets it. The other soil amendment that we're going to look at is what we call a granulated compost. Okay, so this is effectively you might call it a yuppified chook manure. It's very, very lovely stuff. It's the stuff that's on the crate in there. It's made by a very progressive fertiliser company called Terra Firma. As I said, it's a chook poo pelletised compost product. It has a lot of carbon in it, and it has organic forms of nutrients as well. Our idea with including this in the experiment was to see, does this benefit the plant directly? Does it also, in combination with the native soil, does it provide a source of carbon to stimulate growth of that microbial community? And can that benefit the trees in the short and the long term? So we're interested in both the separate effects and the combined effects of these two soil amendments. Okay, so that's the crux. If we just come out and have a look at this plot,